Dear Associated Professor Barıs and esteemed participants, a warm welcome to Bioinfocongress VII. We are truly honored to have Associated Professor Barıs with us today. Workshop training on virus bioinformatics, an area of profound importance and intrigue. Following the workshop training, we'll have an engaging question and answer session, where we'll address both questions submitted by our participants and those thoughtfully prepared by our team. We look forward to everyone's participation to enrich the dialogue even further. Dr. Suzek, welcome once again.
Thank you very much for the introductions. So welcome all. I guess that was the introduction, so we don't need to say more. So if I'd like to start with a happy anniversary. I was going over the website yesterday. I noticed that 28th March was the first day of foundation for this organization. So it's almost 5 years and 1 day today. So happy anniversary to the foundation and who is involved in establishment of this nice foundation. So before I start, I'd like to thank you to the organization for the kind invitation, to all the participants for sparing their time on Friday at such a late time point, and then to my lab's members at Muğla Sıtkı Koçman University for support, and whatever I'm trying to present today are coming from them. So I'd like to thank all of them. Today I kind of divided the presentation into three sections. So I will start with introduction to viruses, because I like viruses. So I will try to share what I like about viruses with you. From an engineering point of view, by trade, I'm an engineer. I'm a computer engineer, but I've been working on bioinformatics, I guess, roughly now more than 25 years. I started in 1998. So I have seen all the stages. Now I've moved to virus bioinformatics for the last couple of years. So there are interesting things about viruses that I want to share with you. And then the heart of the presentation is about virus bioinformatics resources. So here we will discuss some databases and tools. Obviously, the time frame we have today will not be enough to go line by line and ask you to test them step by step, but I will illustrate some exemplary resources so that actually you get an idea on what can be done from or after this session, and then maybe follow up with me if you have more questions. And finally, within the paradigm of virus bioinformatics, what I am doing actually, what problems and solutions I provided, I will give one example to the problem statement and solution I proposed within the context of this presentation. So now this is a part probably majority of this audience know more than me, but the viruses in a nutshell is mobile genetic elements that codes for proteins but do not have ribosomes. So they're kind of like parasitic life forms. They can only survive within a cell. Only if they can infect a cell, they can survive. But when they get into the cell, they can replicate themselves. And this cell, or the owner of the cell, we call host organism. They may contain either DNA or RNA in different forms. They may contain a protein code, we call capsid, or sometimes a lipid protein. We call envelope. So a lipid we call envelope. Now they can be in different types. There are helical ones, polyhedral ones, spherical ones, complex ones. Complex is a catch-all situation that covers many things. But one of the things that attracted me about the viruses is their shape, actually. So they are very nicely organized in terms of like optimization from a engineering point of view. So how big is this world of viruses? There's an organization called International Committee on Taxonomy of Viruses who decides on taxa of viruses. It essentially decides the realms, kingdoms, phylums, classes, orders, families, genuses, up until species, they identify the viruses as they emerge, but they don't get into the details of genotypes, serotypes, isolates, anything below species, they do not capture. The reason for that, actually, the bottom part, the genotypes, serotypes, and these isolates are awfully hard to do anyways. But at species level, that can be done in a probably manageable fashion. Now, this organization is around since 1971, I suppose. They started with 200 or 300 virus species at the time. And then right now we have roughly 16,000 species of viruses. The interesting thing is there is a kind of steep increase in the last couple of years. Part of the reason probably is the COVID pandemic, but the interest in viruses is ever increasing since 2010-ish. Now, and in the last 5 years, the increase was more than twofold in terms of identified species. The virus is considered the most abundant and diverse biological entities, biological being in quotations because they cannot survive alone, but it's estimated there are 10 to the 31 particles on Earth, and by far there is no close competition to them in terms of their existence in the world. So they are abundant. Now they are abundant, and speaking to that taxonomy below species, they have adaptations, or they change themselves quite a bit actually. There are two terms when it comes to viruses. One of them is called genetic drift. So they can randomly mutate themselves, or random mutations occur during replication errors, or nucleic acid can be damaged, or cellular editing through the cell host organisms can happen. They can drift. That even actually creates a slightly different organism. And then there is genetic shift. They can recombine. Recombination is an event that can happen in viruses, or they can reassortment. So some viruses have these segments of genetic material, and these segments can shuffle. One instance you can see to the right of my slide, to the probably left of your slide. Reassortment is a case where if a host is infected by the same type of virus, the two viruses, which probably have slightly different genetic material, can shuffle their segments and come up with a new version of the virus. So this is the assortment. The recombination on the other end, actually during the replication process, replication process, there's a crossover, just like the crossover we may have in our eukaryotic organisms. And this crossover modifies some of the segments, and that is recombination. So through this, actually, their variety also increases. If you look at the mutation rates in different types of viruses, I said DNA and potentially RNA can be their genetic material. Compared to higher order organisms like eukaryotes and bacteria, that's the top right corner of my presentation, they mutate quite a bit actually. Especially RNA viruses have high mutation rates compared to other viruses and far from their closest competitor, higher order organisms, and eukaryotes doesn't mutate this much. So they can change quite a bit. And that's the portion that affects the taxonomy lower than species. All right, so what is the known virus world? So what do we know? How large is what we know? So I checked NCBI virus database. This NCBI is National Center for Biotechnology Information. This is a US-based organization under NIH, National Library of Medicine. According to their numbers as of yesterday, so we have roughly 222,000 viruses. And actually, this is not at the species level. This is at any level. This includes serotypes, isolates, any kind of virus. And 162,000 of them are annotated complete, genomes. Here, annotation is referring to identified genes. So they call the gene boundaries on them, and they call the genes. So the number is still minuscule compared to the overall size of the world, virus world. From a structure point of view, so that was sequence world. If you look at the structures, in a totality of 2046 virus structures exist in protein data bank right now. All of them are nice looking structures. So this is the full virus virion structure. So they solved the full structure either through EM or crystallography in the old days. And if you look at the records, actually they kind of illustrate the complex makeup of the viruses. So it's not like one protein we're talking about, it's a complex of proteins they solved in this case. But again, 2000 number is not a large chunk compared to what we see in our real world. Open parenthesis actually, to my recollection, the predictive algorithms like AlphaFold does not include viral structures either. So we don't have any solution for them in the AI world either. So who hosts these viruses? The hosts, again, I mean, in this context, host means whoever harbors the viruses and where they replicate, who they infect in some sense. Plants have viruses, invertebrates have viruses, vertebrates have viruses, fungi have viruses, bacteria have viruses. Pretty much any living organism have its own virus family that infects it. But on the top of that, some viruses can infect more families, more than 2, sometimes 3 families. For instance, Carmotetraviridea and Nodaviridea are very special families that can infect plants, vertebrates, and invertebrates. So they have members that can infect all 3. So kind of spanning all the families. And between the boundaries and invertebrates and vertebrates, or plants and invertebrates, or plants and vertebrates, we also have, well, actually plants and vertebrates exception, but we have some that falls into the boundaries of different families also. So they can change their host in sense. Right, so in terms of human, I mean, majority of the deal probably is around human. The human viriome, so viriome is referring to the case where there is a bunch of, just like other microbes, there's a bunch of viruses that lives with us, not necessarily pathogenic, they don't harm us, but we are kind of in a symbiotic life form, I suppose. They do something good for us, maybe they may do something bad later on, but they are dormant at the moment, but they live with us. So it's estimated that there are 10 to the 13, yeah, that's a large number, particles per human individual exists, and they have a great heterogeneity. So there is a humongous variety within human viriome. So we have a lot of viruses in ourselves. And it's estimated that there are, in the mammalian hosts, there are 320,000 types of viruses. So there are a lot of viruses in mammals in total. Now, the interesting viruses typically are the ones that have this event called spillover or host switch. These are particularly dangerous for our health because when they switch to host or to spillover, our immune system somehow do not have the necessary means to fight with it. So we got pretty bad sicknesses out of them. And this is not a rare event, that rare an event, because we all see this on a yearly basis. Some virus decides to jump from a bird to human, and then that infection, if it results in an epidemic or pandemic, we suffer a lot. So here in this picture, we have the influenza viruses that apparently can share hosts, and there is a pig man in the middle. So there are viruses that both infect pig and man in the influenza family. So that's the kind of section where veterinary sciences and the life sciences, in our case, or actually clinical sciences, intersect. How do they look like? I said one of the things that I like is they look nice. They come in all sizes, actually. There's parvovirus, which is 20 nanometer, and there are viruses like 360 nanometers, quite large. And then the Ebola virus kind of has an interesting structure, again, a large virus. So you see in this picture, actually, there's no one distinct structure for them. I highlighted adenovirus here. It's 90 nanometers. It's a small virus, but I highlighted that because in the second part of my talk, or last part of the talk, I will come to adenoviruses again, but they come in all shapes. So going back to the genetic material they contain, so we have DNA viruses and RNA viruses. Within DNA viruses, we can have double-stranded DNA and then single-stranded DNA. And within RNA viruses, we can have double-stranded, single-stranded, negative and positive strands. So both exist. And on the top of that, they can have linear and circular genetic material structure. So pretty much these L and C letters you see in the screen are referring to circular or linear representation of the genetic material that these different viruses have. And then to make things a little bit more complicated, they have this envelope structure. So if you pay attention to the bottom left to my, maybe right to your, some viruses have, all viruses have capsid material. This is kind of like an encapsulation of the genetic material, but some viruses on the top of it has another lipid envelope, and that creates another taxonomy or categorization between the viruses. Those have envelopes and those do not have envelopes. Here, for instance, adenovirus is a DNA virus. It's a double-stranded DNA virus, and on the top of that, it is non-enveloped. So it's kind of one way of looking at the taxonomy of the viruses. So all kinds of genetic material as well. So what do they do in human? So when we host them, so as I said, actually, we have different types of viruses. So we have eukaryotic viruses. So these are the viruses we should have because we are eukaryotes. It's normally we should have. And then there are phys as well. So these are the kind of viruses specifically infect bacteria under normal circumstances. So if we start from far right, the phys that we harbor somehow believed to activate immune responses somehow within human. They can regulate bacterial abundance and composition. So somehow they either suppress certain bacterial population or kill certain bacterial population. So they are regulating our bacterial composition. They can transfer DNA and somehow affect the pathogenicity and fitness of the bacteria. So these are considered kind of maybe beneficial to us properties of phys that we harbor. And then going from in the clockwise direction, so they can help us to, by simulating immune response, can help us to prevent other viral infections, the ones that we have, and then can replace the beneficial function of, so these are kind of maybe bad things. So they can replace the beneficial function of a bacteria if they infect the bacteria or transfer their material. So that is what we don't know. But when we reach to the top left corner, they can induce chronic infections is one thing of importance. So what kind of infections they can do? They can cause respiratory system infections, they can do immune system infections, digestive system infections, nervous system infections, and then any kind of, I guess, skin-like infections. So they have, I mean, if you look at the type of infections they can cause, there's also a vastness and richness when it comes to their capability in terms of infection. But note that they are also beneficial to some extent. So it's good at that. So why are viruses important? They are not only pathogens, of course. So like the corona we have seen, the simple influenza they cause, the simplex, herpes simplex in human, but they also infect plants. So they are not that friendly to friends either. So they can cause different diseases in plants. Cattle, livestock, they can infect as well. I mean, not necessarily the same ones, but there's a wide variety anyway. So one of them infect. So they have economic implications, health implications, social implications. So there is a lot of problems they cause. So this is their pathogenic side. But that's not the only side out there. They are very interesting biotechnology tools as well. So, so of course, to be able to develop vaccines, you need them because you are creating vaccines against them. So you need to have them in order to do vaccines. So this is the therapeutic aspect, one of the therapeutic aspects of them. They are almost natural vectors for gene therapy. I mean, a virus naturally gets into human body, reaches out to the cells, gets into the cell, replicates itself. So it's naturally actually a kind of like an encapsulation of a drug or gene. So seeing this opportunity, they use them as vectors for gene therapy if they want to deliver a genetic material into the cell. They use viruses as vehicles. The bottom left corner, I was not aware of that before this presentation, and I kind of thanked you about that. Now, viral proteins are the type of proteins our immune system somehow do not react against. So if you have any proteins that are not naturally available to us through some rare disease in our body, if there's a viral version of that protein, and that protein happened to be a functional protein, and it's quite remotely homologous to our version of the protein, somehow enough to not attract our immune system, they are used as replacements of our protein. So that's kind of, I guess, new area. So protein, their viral proteins have therapeutic impact. And then on the bottom right corner, it's kind of one of my research interests, oncolytic virotherapy. Now, a virus normally gets into the cell after replication and after saturation. All the resources a cell can offer to the virus, it bursts the cell and leaves it. So it kills the cell. So oncolytic virotherapy uses either this mechanism of action, like bursting the cells, or they can deliver certain material into the tumor cells and become an oncolytic therapeutic. So viruses have this oncology therapy, or I mean cancer therapy aspect as well. If you check clinicaltrials.gov, which is the US-based clinical trial registration side, you will see actually there's a decent increase in oncolytic viral therapies in recent years. So there is, especially in certain viruses, they are getting into the at least second phases of the trial, and there are actually drugs that are already in the market which are using viruses, not the gene therapy ones, but for oncolytic therapy. So they have good side as well. So before I am completing this part of my talk, I just want to say viruses are interesting because of their vastness and variety. So this vastness and variety opens up a lot of doors. If we don't know something today, tomorrow they may emerge as a, let's say, a protein resource, a new one, or a potential oncolytic therapeutic agent, or maybe something that will be used in some biotechnology tool, biotechnological use that we cannot imagine right now. So there are, because of this variety and vastness, there are a lot of opportunities. We know they have benefits and harms, but learning harms and trying to coping with them is one thing we need to do. But again, going back to what I said earlier, finding their benefits is an opportunity. Still being explored territory, so it's kind of tip of the iceberg what we see. Probably there's more to come, and we will see more in the future years. So it's a large area that needs to be explored. Again, emphasizing it has significant potential in future therapies because these not biological entities have natural skills to evade immune responses and kill host cells or kill cells. So they are very powerful in that and very efficient in that. They do this with minuscule genetic material with using cells' own factory to survive. So yet there are some challenges on studying them. So some of you may say, wow, it's interesting to study viruses, but there is, of course, these viruses, because of their harms, require certain treatment when it comes to wet labs. So, for instance, if you are dealing with lethal human disease viruses, you need to have a biosafety level 4 lab, or anything serious or lethal human disease biosafety level 3, and the risk goes lower as the lethality decreases. But these labs are not only expensive, but also scarce and require certain training. So on the right-hand side, the map shows actually the reds in the map actually are biosafety level 4 labs in the world. Or I didn't capture the world here, only Europe and us in Turkey. According to the records here, we don't have any biosafety level 4 lab, for instance, if you want to study anything dangerous. But in Europe, there are one in every country on average. But some countries don't even have it. So if you plan to do some validation studies or some clinical work or some trials with viruses, these biosafety levels become important to observe or monitor. So there are these challenges in studying viruses. Right. So with that, I will shift the gear to the virus bioinformatics resources. So I will try to tell some story or motivation for each slide. So why do they have this? This, again, is going to be an exemplar set of bioinformatics resources, not necessarily covering everything. So I will provide list of URLs in the slides. They are in the slides, so you can explore them post-presentation if you are interested in working. So I divide it into two. So one of them is databases, resources, and then the other one is tools. So we will discuss them kind of separate. When we refer to database and resources, I mean, after all, viruses are just biological entities, and we already have general purpose bioinformatics resources out there. If you are a little bit familiar with bioinformatics resources, for instance, for protein-centric information, there is Universal Protein Knowledge Base, or short UniProt, that you can essentially access any kind of protein information regardless of the species. But UniProt also serves viral families. Likewise, NCBI or EBI's GenBank or NCBI's RefSeq are nucleotide and protein resources, respectively, and not respectively. So you can either access nucleotide sequences or protein sequences. Again, these are generic resources. They don't really mind about organisms, but they also have viral proteins and nucleotides, or UCSC, genome browser. And pretty much if a site or its resource is presenting or posing itself as a resource that's serving any organism, they probably cover viruses in them. So that's the general purpose one. But this presentation, I will kind of focus on virus-specific resources. So if we narrow it down onto viruses, what is available out there? So that's we are going to detail. Again, I said, if you're interested in about these resources, there is not that long ago a publication that kind of collaged these virus databases in 2023. Yet, actually, I noticed that some of the databases are no longer supported. So you need to be like picky about which databases you are going to choose, but you can go to that paper and find what database is available and visit after my talk. Also, the European Virus Bioinformatics Center hosts a list of virus bioinformatic tools and also databases. The link is here. Again, there's no guarantee all of them will be available to you. And in my presentation, I will also highlight some. But if they are not available, the purpose they are serving probably is served through another resource. So you can find the alternative resource that emerged to fill that gap, at least gives you a pointer to the right resource. So with that, let's start with our first resource. This resource called this Viral Zone. It's a Expasy hosted, so it's in Switzerland, Expasy hosted site. It describes itself as a web resource for all viral genre and families. They provide information. So this is not like a nucleic acid database or a protein database. They provide kind of encyclopedic information about viruses, and they present nice figures. So links again are available in my slides. If you open up their site, you will see actually they have this nice taxonomy presented at the top of the site. There's search capabilities. They have, I mean, if you want to contact them, you can contact them. So I highlighted in red on the top left corner. It helps you to navigate their resources through classifications by hosts, by different hosts, and based on the hosts of the viruses like human, bacteria, or based on the interactions. And just to illustrate, another thing for you is to highlight is they have updated this database in 2025. So it's an active resource. So one thing you might want to pay attention when you are visiting a resource in general in bioinformatics, you should check whether their release is recent. So whether it's updated or not, because historically in bioinformatics, because it requires some resources to facilitate or handle these resources, you need also funding to support them. Sometimes funding runs out and they decide to abolish the resources. So not to experience that kind of bad taste, just check their updates or release time. So try to make them, try to see if they are recent or not. Anyway, so if you follow their navigation, so these are, I just looked for like human viruses, and I specifically want to highlight that what kind of information they offer to us. So of course they come up with the taxonomic information about viruses, the hosts they capture themselves. So these hosts later on I will highlight again, but typically hard to create, and the hosts require some human intervention, maybe reading the literature sometimes, typically not that easy to capture in viruses. So they put some effort to create their hosts. They report epidemiology incidents, like if has there been an outbreak, an endemic, a seasonal endemic, all those things are reported. They report their prevalence in terms of worldwide, their pathology, so the fatality rate. So all these information under normal circumstances may need you to, or may require you to do a lot of literature review, already created and provided through Viral Zone to you for virus families. Now, on the top of that, they have pretty decent description of virus families. So, for instance, here in Adresimizden, they have the virion structure. So, in this case, there is a capsid, and on the capsid there are these fiber proteins, exon proteins. I mean, all those details are captured, and these illustrations actually are, I think, to my recollection, kind of scientific commons license, so you can use them, and you can navigate, and it highlights it. So these are vectoral graphics. So, and there is a lot of information embedded here. It tells what kind of strains it has, what's its taxonomy, and all those details. It's kind of like opening up a dictionary and reading about a virus. On the top of that, on top right corner, they have links to the databases for this family. So they have, some of them I will show shortly, but they have links to NCBI virus, for instance, Uniprot. So if you click them, you will see it will take you to all the proteins, for instance, in the case of Uniprot, that belongs to Adenoviridae family. So all the proteins from that family, or NCBI virus will pull out all the viruses from this family, kind of idea. Right, so the other resource, kind of, we are going with the kind of central virus resources, Bacterial and Viral Bioinformatics Resource Center. Here, they have bacteria, but our interest in is viruses, so it's kind of Microbial Bioinformatics Resource Center. It has, it's trying to support infectious disease research. Again, they integrated a lot of data resources for you. I provided their link. I mean, they have not only data, but they also have some analysis tools. So now you might say, what are these analysis tools? These analysis tools are no different than the ones we see for, let's say, eukaryotes. They have genome assembly tools. Typically, for higher order organisms, you cannot assemble a genome using an interactive screen on a website. But because the size of the virus genomes or genetic material are small, they allow you to upload the reads of your, let's say, sequencing project, and help you to assemble the full virus genome using their assembly pipeline implemented through their website. So you can do that. They can help you to annotate, so assign function or name to your proteins. They can help you to BLAST, meaning conduct sequence similarity search using BLAST. They also have the facilities to do multiple sequence alignment. So they don't only provide data like genomes, proteins, and genes, or the distribution, but they also provide some analysis utilities in a central fashion. So on the top of that, I pretty decently updated, actually. They report on outbreaks involving viral or bacterial bacterial species. So here, monkeypox and influenza H5N1 outbreaks are reported because it involves viruses. In this context, they report them. So it's, again, just like viral zone, a central resource one can use when it comes to viruses. If we use their search facility, let's say we want to do protein search. I artificially conducted one here within the Adenoviridae family. I looked for host human, the family members that are from human. And then when I search it, it gets me a list of genomes that are for human host. And then this is the part where viruses become complicated, because I mean, minuscule changes in their genome lead to different species, or I mean, lower levels of species, lower than species. So we have all types of viruses that are infecting human, that's from that family. They provide the genome size, all those, I mean, the kind of proteins. Sorry, in this context, they are providing the proteins like hexone protein. I mean, there are other proteins listed here, but they also have this on the right-hand side. I highlighted that way to jump from here to other resources of your interest. So there is a NCBI gene bank accession here, for instance. So from this, you can jump to their representation of the identical sequence. When you are traversing this, you can go to there and also look what they have in about this particular protein. So in that sense, it's integrated with other resources. Right, so moving with this same idea of central resources, we have NCBI virus. You know, NCBI has a lot of databases, and actually NCBI virus is a derivation of RefSeq and GenBank. So it's using the sequence databases they already have within NCBI, and they are filtering out viruses out of it and presenting itself as a as a alternative resource. It's kind of a view to existing sequence databases. So though it provides its own search functionality and sequence similarity functionality, so you can in this screen search only against virus genomes or nucleotides or protein sequences, and you can search only viruses. So it kind of excludes any other organism from the view. So in that sense, it's kind of right to the target. So what happens if you search something? Here again, I searched influenza. Now influenza, when I search influenza, obviously there are many influenza genomes available. So the nucleotide is referring to different nucleotides that come from that organism, influenza. Now you can also pull out the proteins or virus assemblies to the genome. So all are in the screen. On the top left corner, you see different filters. So here we are in the gene bank sequences filter. But if I shift it to the visual filters, for instance, the visual filters is referring to a view where you can see where these individual isolates or the nucleotide sequences come from internationally. So they report when they are collected and the individual release times, countries, I mean, a lot of information about the location, geographic distribution of the extracts. And then they also report outbreaks. And in the context of outbreak, and if you pay attention to the top right, bottom right, and you will see the for a particular day and week, they have identified an outbreak, and they have, in this case, let's say 8 + 8, a total of 8 sites, U.S. territories, and Ohio and Colorado, I suppose, 2 places. They have 4 genomes collected, and then they also collected, probably it's an international, so within U.S. 16, and then outside U.S. 8 more in Germany. So they identified a set of viruses that came from these locations. So not only the outbreak information, but also the samples they collected from that outbreak can also be accessible through this site. So that's a rich resource if you are really working on outbreaks and phylogeneity of the viruses involved in the outbreak. So this database is pretty new, and I think it's meeting a need. So for a while, we were looking for orthologous proteins from different virus species of the same family, an authoritative resource. And we were computing through sequence similarity ourselves, the orthologous groups. But this group came out with a database that's on a I think 2 weekly or 2 monthly basis updates their orthologous groups, meaning, let's say for the, I don't know, virus, if you are dealing with fiber protein, they have fiber protein orthologous groups that you can group. Now, and then the other, they also have this hierarchy of orthologies, starting from the most detailed one, as in like specific one, and then it goes higher in the ladder, and as you go up in the ladder, there are the remote homologous or remotely orthologous sequences are grouped together. So there's a hierarchy in their orthologous families. Again, it's publicly available, but unlike other databases, this so far do not provide the information in a web searchable fashion, but rather they provide simple FTP site that you can download the information, and you need to parse out or use some scripting to extract the information, though the documentation is rich, so you can do it. If you are dealing with virus bioinformatics, the orthologous families are quite handy in reality. Right, so another big problem I faced is actually finding the hosts for viruses. Now, if you look at the records in different databases, in nucleotide or protein databases, you will see actually they typically write down the host or a host in either literature provided in a note, not necessarily in a computable format you can automatically extract and maybe assign a taxonomy identifier to. So it's not that easy to normalize it or extract it. So these guys in Japan went over and did it actually for RefSeq Engine Bank and Unprot Viral Zone on literature. So they collected host information and created quite a bit of viruses and their hosts, and they provided through a website. So this site, if you go, tells you for different virus families what are the hosts of individual adenoviruses. Here on the red, you see a adenovirus, beta adenovirus 2. Now you might say, actually, Dr. Suzek, we all know actually it's bats, but simple bats even have 2 different species, and you might in reality need to know which one or which one not it's infecting. In this case, apparently the literature or wherever they created it from reporting only 2 bat species, not necessarily all the possible bats out there. So this is a crucial information. So in that sense, it's a useful resource. And for all of their identification, they provide some citation. So they either cite the literature or they provide a database reference. Right, so these are primarily either the taxonomy, sequence. Now, if we look at interaction databases, so there are also interactions involved. String is again, general purpose protein-protein interaction database. So probably because of the COVID or because of the influenza outbreaks, they come up with a view of virus host and virus interactions. So in this view or database, they provide, just like string itself, interactions, physical interactions created from databases or experimental determined, or the special interactions like gene neighborhood, gene fusion, gene co-occurrence, or other kind of interactions like referring to text mining, co-expression, and protein homology. So the one thing you need to be a little bit careful about is the interactions here are not referring to physical interactions, just like string. These are like more like relations between biological entities. So if you go to their site, it operates similar to string itself. You provide, though, a little bit different in the sense that you provide the virus itself and the host, for instance, in this view. So for hepatitis B virus in this example and Homo sapiens as the host, if you search, you retrieve a network of proteins. Some of these proteins belong to hepatitis B virus genotype C, and then some of them, the gray ones, are belonging to Homo sapiens, and they highlight these edges that are highlighting the interactions between these 2 proteins. Right, so in terms of resources, the last resources I want to highlight is something called European Virus Bioinformatics Centers. The idea here is not necessarily to provide a database, but a platform, a resource to facilitate interaction between virus bioinformaticians. So whoever interested in virus bioinformatics as scientists can come to here, maybe reach to training courses, maybe reach to other collaborators, maybe advertise their tools. So it's a site, kind of like a social media site for virus bioinformaticians. So their site is in Germany. If you go to their site again, it has social aspects, as in there are events, workshops, advertisement of the tools, upcoming conferences, seminars, the sub-publications. Potentially you can become a member. They offer lectures, so you can attend lectures. So there are all kinds of resources they offer from one site. Right, so that was the resources and the databases. Now on the tool side, the tools, of course, are in general no different. Some of the tools are no different than other tools that are offered to other species. So if you want to do sequence assembly, you can potentially use other sequence assembler for, although they are tiny, but various genome assemblies. BLAST is still the same BLAST. After all, this is nucleotide sequence, or they have protein sequences. When they translate it, same BLAST will do it. Any pairwise alignment tool will work for viruses as well. Or if you are doing multiple sequence alignments, they still hold the ground, so they work. But there are some virus-specific tools, like those for genotyping, phylogenetic analysis, protein-protein interaction, prediction in the context of viruses. So like niche products that are available only for viruses. Again, not comprehensive, but a pretty decent list of them are available through the European Bioinformatics, various Bioinformatics Center site. They are updating their tool list, so you can find the others that I cannot talk about today over there and see if you can use them. So the first resource I need to talk about is called NextStrain. NextStrain. NextStrain is a set of open source tools that helps us to epidemiological understanding of pathogen spread. So because these viruses are pathogenic, and then through their lifetime, they also evolve. So there are tools in need for their tracking their spread and evolution, and then maybe as a result improve the outbreak responses. So they brought together a set of tools. I'll go over the tools. Agur is a tool that does phylogenetic analysis for you. This is not a visually appealing tool, but it does run a pipeline for you. That's in essence. ASPICE is an interactive tool for visualization of phylogenomic data. And then finally, NextClad is a tool for kind of doing genome alignment, mutation code, and clads as in like subtype or anything below subspecies level assignment. It's open source, so you can use it anytime. Their websites kind of have example analysis for you if you don't plan to do your own analysis, and actually even examples are compelling. They have a nice documentation, so they have tutorials. You can run tutorials. Now, what you see is ASPICE that tells you many things in one screen. So this is a avian flu data set. What you see here is how avian flu propagated to different species. So in phylogeneity, they highlighted the blue as avian, and then orange as cattle, or there is human as well. Note that they collected different specimens in different times from different individuals or animals, I suppose, and they superimposed or projected this to this graph, and you can play this graph within a time range. And you can also show this on a map, actually, so where all this data is collected from. And on the top of that, you can illustrate the nucleotide diversity of the genome. So where it changed a lot or where it stayed the same. So in one screen, they kind of summarize the outbreak or ongoing outbreak of influenza in North America, geography information, phylogenetic information, the variant information summarized in one screen. Right, so the other application, NextClad, is again, in this case, let's say you are collecting specimens from people who have been infected by influenza. And then you collect the specimens, you sequence them. Now you have genomes of these specimens. You assemble them and you have the genomes in hand. So you can upload these genomes and somehow it can suggest you what kind of organism or reference data set it will compare it against. So for the purpose of this exercise, I artificially, synthetically generated SARS genomes and then uploaded them, and it automatically recognized that this is probably SARS-CoV-2. So, and then when I run the analysis, it gives so the sequence name is essentially on the second column is my own sequence, whatever the collected specimen or name I gave the collected specimen. It computes its differences in reference to the reference genome provided. In this case, is SARS-CoV-2. So it also highlights the variation, the differences, similarities of that against the reference for all of them. And then I can also draw phylogenetic trees using my collected specimens and full sequence. So I can create trees, phylogenetic trees out of it. And I can, of course, export them, visualize them. So it's a decent visualization tool. So this is the ASPICE tool. Note that actually the first screen you saw had the geographical locations as well. You can think this as a dashboard that brings together these tools together, the first screen. This one is just one portion of that screen. So phylogenetic and geographical location analysis. There are also tools for genotyping. So here you have a sequence, you want to assess the genotype, what kind of subtype this virus you have. There is a virus genotyping tool site. Now, unfortunately, at the moment, this is not fully operational, so many of the things are dead-end, but there is a certain need for this one. Here, for instance, there is a subtyping tool for HIV-1. So you take your HIV-1 genome sequence, you paste it, and then the tool predicts actually what kind of subtype your HIV-1 sequence have, and then it reports it. So it's a predictive tool, but it's a useful. So as I said, it's not functional yet, but the need is there. So you need to do genotyping for some viruses. There are occasional need for, of course, assembling the sequences. So let's say you have your own facility and you can sequence viral genomes, and viral genomes are sequenceable. I mean, they are not as large as eukaryote genomes, so you can sequence them probably on a daily basis or hourly basis with the sophisticated technologies we have. But you need a bioinformatics pipeline to do some of the things for you. So this pipeline, vPipe, developed by a Zurich group, again, takes your read data that comes from the sequencing machine, viral sequencing experiment, in short. And then as an output, of course, it creates the quality control, the read alignments. It also infers the viral genomic diversity you might have. So although you might be able to do this pipeline from scratch using the tools that are on the shelf, but because this tool has been used by other cases in the world and has been cited quite a bit right now, you can simply cite, I used vPipe in my analysis, and it will gain some credibility. So it's a, again, open source, freely available pipeline. The last example I have under the toolset is HVPPI. It's just to illustrate, there is also there is a set of tools that predicts the interactions between human and virus proteins. So one human protein, virus protein, which is a critical interaction for infection or endostasis, essentially in virus entering to a host cell. So predicting that relationship. So there's one tool, again, many tools are in existence, but these tools typically use some kind of machine learning approach, or you may say AI. What they have is some numerical representation of respective sequences. You paste your sequences into them, one from host, one from the virus. In this case, you need to know what kind of capsid protein or envelope protein binds to a potential receptor on the host side. So what kind of interactions you're expecting should be known to you. So you can run this and it will tell you, yes, they will interact, or no, they will not interact. Or sometimes they assign a probability to their predictions. So they may say by 75% probability they are interacting, by 25%, which is its error range, they are not interacting. So this is the other set of tools that we use in various bioinformatics. Right, so before I shift to the last topic in my talk, I think I am a little bit ahead of schedule right now, but that's okay. We can have a longer session at the end. As I try to present here, many resources exist, and some of them are disappearing, and many more are being developed in parallel, because if there is need, there are new tools going to be developed. But there is enough, like probably compared to a couple of years ago, there is enough out there to conduct mainstream research. So if you are doing phylogenetic using viral strains or specimens, there are a lot of tools out there. If you want to sequence, assemble, or decide on diversity you have in your hand in terms of virome, there are tools out there. Though not all of them, in my case, actually, protein-protein interaction tools are not working perfectly, but as years pass, probably they will get better. That's the page for more probably needed, and better ones in some cases are needed. Obviously, even for some, I don't have the full expertise to use them effectively, but they require some level of virus genetics knowledge and some data that you can feed into these tools, or ability to search the databases. So that calls for expertise as well. So these are needed, and probably this is also a prerequisite for majority of the resources outside. So you cannot, as a high school student, get and start using these tools for sure. But I think the audience here is a well fit well for the use of these tools. Right. So last part of my talk is a little bit on my perspective. So what I did that brought me into the virus bioinformatics. Because we are doing well in terms of time, maybe I can tell a bit of a story. So the reason I got into virus bioinformatics, actually, I had a PhD in my PhD committee back in George Mason University. There was a person who was an expert in adenoviruses. So he's a world-leading expert in Adenoviridae family. And at the time, I did a rotation in his lab, and we were trying to fix the genomes of adenoviruses So one take-home lesson from that experience I had was the virus data is noisy and dirty. So I did quite a bit of work to clean up the adenovirus genomes in his lab that were already deposited to GenBank. So even GenBank didn't, clean them up when they are entering them into the database. They are just sequences, but either annotations were wrong or they were partial. So I did that problem. So Don, his name, at one point told me we should do a project on viruses. So that's how I started to get interested in viruses. But once you get it, actually, if you like viruses, it's an interesting field. Now, one thing to keep in mind is actually probably viruses is not going to be more in our lives in upcoming years. So one factor affecting this is whether it does exist or not there is a contribution of climate or the climate change somehow are increasing our exposure to the viral strains that we haven't seen before. For instance, the glaciers, as they melt, some of them being 15,000-year-old, the frozen either air bubbles or it could be liquids somehow contain some load of viruses that we will be exposed to. Or like lakes, because of the ice melting, will have additional virus loads. Or because of the urbanization in rainforests, we are exposing to new animals that we have not encountered before. Those animals probably are loaded with new loads of viruses that we haven't seen before. So this climate change, urbanization, or environmental effect that we will face in upcoming years. Now, illustrated in a case where simple things, environmental things leading to disastrous conditions. So the Hunter pulmonary syndrome is a disease probably lethal for some, emerged as a follow-up to a El Nino southern oscillation. So this is a weather event which increased the precipitation in a certain location, which resulted in increasing the production of certain nuts, which happened to be a food source for a mouse type, deer mouse. And these deer mouse has the hantavirus in their droppings. So, and these droppings, because of the excessive populations of the deer mouse, contaminated floors and blankets, which eventually reached to human, and then human got sick, is the thing. So a weather event leading to a new virus exposure. The other thing we are seeing is the spillover events, and probably COVID was one of them. We saw this in the past in, maybe some of you know or not, there were other influenza viruses that were spilled over from other organisms. When this spillover event happens, I mean, actually HIV considered a spillover, or canine parvovirus that affects dogs, but still a spillover from other organisms. So there is this event happening, and more probably will happen because viruses have this tendency to mutate and adapt to different hosts. So if we visit the factors affecting this, the mucosal surfaces is contributing to potential spillover events. Receptors are contributing to these events. The protein modifications affecting IFN immune responses are contributing to a spillover event. The population structures or the transmission processes, I mean, there's also a social aspect of transmission, of course, because large families living in society have a contributing factor to potential spillovers, or hunting, as simple as hunting, may be a contributing factor to spillover. The virus's evolution, which is natural to it, is contributing to spillover events. So out of all these, I picked receptors. So we asked these questions. So can we predict a virus's host or hosts? Or if there are most hosts involved, can we predict spillover events? So again, this diagram in timeline from 1997 to 2012 is highlighting spillover events. So as you say, pretty much every year, we are seeing at least one incident. So it's happening. Some of them are more dangerous than others. So 2 questions. Now, to be able to solve these 2 questions, we cannot obviously attack all the virus families. So we picked one. So I went to my advisor and I said, can we do adenovirus as a model? So adenoviruses, of all the viruses out there, is one of the well-studied ones because it's infectious. It causes infections, and it also is a vector for gene therapy. So there is a community working on adenovirus family. So that in that sense, actually, it's a well-studied organism, biological entity. It's a 90 nanometer virus with genome size ranging from 26K to 50K base pairs, small. It has a capsid, no envelope. In the capsid, there is this fiber protein which anchors itself to capsid through penton base. But this fiber protein and its knob is the critical component for the, on the right-hand side, you see more clearly, for the adenovirus to bind to a receptor and then through endocytosis get into the cell. So the critical portion for virus to be able to infect or evade a cell is the receptor binding. So, and luckily, the serotypes or the variety of the adenoviruses studied is quite large. So we needed data. So adenoviruses kind of fit to our purposes when we were doing this research. So now we revised the problem. We identified the model organism. So we said, using receptors this time. So I am kind of excluding all the other contributing factors. So it's an assumption. Here's my engineering hat. I'm wearing my engineering hat, so I'm simplifying the problem. Using receptors only, can we predict hosts of an adenovirus? Or can we predict adenoviral spillover? So now I have the model and a scope for my research. So in terms of methodology, we did a literature review. Again, it's not like everything served you in silver plate or in the virus world. If you go to the databases, they only offer you a few of the receptors, but we needed all the receptors to be sure on our analysis. Even in current form, we may not have all the receptors identified, but at least we did spent quite a bit of time to identify what kind of receptors there may be on the human side that may be attracting adenoviruses. So we created the 8 receptors. And then we went to the complete genomes and for our adenoviruses identified 40 different hosts with complete genomes. So the reason we picked complete genomes is we want to make sure actually all these 8 receptors, if they exist in a given organism, is in there. So we made, a receptor may not exist in a potentially in an organism, but if it is complete genome, if it is not there, it's not there. But if it's a partial genome, we wouldn't know. So we made sure actually everything is done on the complete genome. And 75% of these hosts were mammalian and only 25% were primates. So that's the distribution you have. So a lot of data creation and collection involved here and cleanup as usual. So then we collected 240 receptor proteins for these 40 hosts. So it turns out our assumption was right. Some receptors do not express themselves in some hosts, which is okay. And then because the fiber protein was the culprit on the other side, we identified 250 adenovirus fiber proteins. So now we collected enough data to come up with a prediction algorithm. Now, we didn't develop our own protein-protein interaction algorithm, so we went to, again, to literature and identified there are 4 at the time. Probably now there are more, but at the time we identified 4 machine learning models to be able to predict virus host protein-protein interaction. So you might ask actually how this is different than regular protein-protein physical interaction. In the virus host case, the coevolution principle does not hold that well. So in, for instance, protein complexes in human, if 2 proteins are interacting in a cell, they should evolve concurrently so that they continue to interact. Otherwise, the cell becomes non-functional. So in the case of virus, which is kind of opportunistic, some variation, some recombination, some some event may all of a sudden create this interaction, and another event may remove the interaction. So it's just depends on the pace of the mutations and its chances. So in that sense, these algorithms probably are capturing that pattern. So with that idea, we took 4 of them. And then this is called in machine learning stacking or meta prediction. We took 4 algorithms prediction for 8 receptors as a vector. We computed this. And then on the top of that, we also included host taxons. It could be species level or it could be like genus level. The reason we did include host interaction, host taxon is typically the question we have is whether it infects a host or not. So we will kind of know what host we are testing it against. So that's available to us. So that actually boosted the performance a little bit, but later on we decided not to use it because it's not generic enough. Now, stepping back, what's the idea here? So let's say one of these days we have the technology to isolate a new virus genome from Adenoviridae family. And we isolate it. We find its fiber protein. And then we want to decide actually whether it will infect human or not. If this model works, we can do it with a certain accuracy. So that way we can probably come up with a precaution against that that type of virus we identified. Anyways, so in these algorithms, you need to have a positive subset, meaning actually the interaction is happening. We know the interaction is happening. Or in this case, actually, we know a virus really is harbored by that host. We know that through the earlier database, actually, at least one of the resources, virus host database. And then the things we don't know, as in like, there is no documentation out there, there's no literature out there, there's no source out there saying this virus is hosted by this particular, harbored by this particular host, is not available. We put it in the negative subset. Now, this is not the perfect negative subset, as in like, it's not validated. So we don't know really in our negative subset whether that negativity is correct or not. But this is the only thing we had. So we did then run a lot of experiments. Before I talk about the results of the prediction, let's talk about what we experienced. So one thing I relearned that is the data available in bioinformatics resources for particular viruses is noisy. So hosts are not annotated. The fiber protein have all kinds of variations in naming. So there's fiber, there's fibre, fiber protein, fiber homolog, protein fiber, fibra protein, fibra homolog. In some cases, hypototical protein, were all kinds of names assigned to fiber protein for a given adenovirus. So we needed to deal with this cleanup. So harmonizing, collecting this data certainly was no picnic. And not that we started the process with creating our own receptors for hosts. So then we run these algorithms. The one trial we did actually, because this was published and one of our referees said actually, did you test the correlation between the prediction algorithms? So we tried 3 algorithms out of 4. One of them we couldn't run local, so we decided to exclude that one. 3 by 3. One thing we saw that the algorithms, although they are available, do not agree on their predictions. So that was kind of funny, but it also opens up a potential opportunity for us. So using a collage of them, ensemble of them, actually probably will boost the performance. If they all agreed in their predictions, the ensembling them or assembling them wouldn't make sense. So we kind of justified using a collection of them rather than using only one of them. At the end of the day, of course, we tried many experiments with many parameters, but we achieved a 88% sensitivity. That is meaning we can predict a virus's hosts correctly in 88% of the cases. And in 83% of the cases, when we say a virus host is wrong, that's really wrong. So the negative set was also predicted correctly. So there is still some level of error here, which is normal. Now, we also did, I do not present it here, but we also did, what if happens actually we use one single receptor to replicate these experiments? It turns out single receptor didn't give the similar performance results. And that also illustrated us there is a receptor preference in adenovirus families. And this we did know, but unfortunately in the literature, there is not that many documentation on which adenovirus serotype uses what receptor to infect an individual cell. So that level of resolution doesn't exist. So, but at least we saw that kind of pattern holds the truth. We checked false positives qualitatively. That's wrong host prediction. So could it be a potential spillover? 15% of the human viruses infected other primates. Now, the human primates spillovers are known incidents, actually. So maybe not all, but some of these 15% are maybe correct, because again, going back to biosafety level and potential expenses involved in validating this information, we don't have a way to validate this or test this. The 6% of them are one primate infecting another primate. Again, primate to primate infections, non-human primate to another non-human primate infections, spillovers are common. And finally, human to domestic animals like dog and cat, we are reported as false positives, but there is literature that because we are habiting the same environment, the spillover between human and dog and cat, whether it causes infection or not, is seen. So that's also potential. Let's say there's a valid point to it. One of the false positives, actually, so we were looking at large false positives, and we find out actually, although at the moment the host was not reported, but there is a species called titi monkey, and it has its own adenovirus. And in the literature, we identified in a research center back in the day a titi monkey adenovirus, which was the adenovirus we were using, and we identified infected human, but it was a false positive based on the host assignments we had at the time. Infected a lot of primates in a research center, and 23 out of the infected titi monkeys, 18 out of the 23 infected titi monkeys died in this mini outbreak. Then, interestingly, a researcher who's working in close contact to this monkey colony, titi monkey colony, also have the same symptoms and get infected. So this is potential spillover. And then on the top of that, one of the members of this researcher females, which have no direct contact with the monkey, also had the same infection. So titi monkeys infected themselves, and then it spilled over to human, and that once it's in human, another human got infected from that human. So it's many jumps involved. So this was kind of like what we probably are catching some spillovers idea emerged from us. So we reported this in our publication. So in conclusion, so this is for this study, we created the receptor data sets, but not only that, plus we created the process to collect it. So in our lab right now, I think we did this for influenza as well, later on, and other viruses, but the other viruses are not well studied as well as adenoviruses, so we need to wait a little bit. For the first time, to our knowledge, to YMS knowledge, we use the receptor-based virus host prediction model. It's developed, tested, kind of worked. But it also illustrated there are opportunities and needs in virus bioinformatics. For instance, there's a need for validated negative sets. So right now, it's really hard to find a data set that tells this virus will not infect this host. But in a lower resolution or higher resolution from the perspective we are looking at, we don't have a data set that tells these two proteins, one from virus and one from human, does not interact. There's a data set called negatome, though it's small. In order to create good machine learning models or AI models in modern terms, we need validated, large negative sets for the purposes of our studies. There is an opportunity to clean the data. As the importance of viruses increase, it's Viral Zone's effort, it's a European Viral Bioinformatics Center's effort. Clean data is in essence, so we need to clean the data. And we are, as we speak, are working on new protein interaction models, because we have seen that none of them, and partly because of the negative sets absence, are working perfectly. So there is that still room for new algorithms out there or models. So I did tell this what we were expecting from this study is, so in the future when the time emerge, so we are hoping that there will be in the mountain, you will isolate a virus, you will sequence it on site, take the genome, identify the virus species, and then through some of the tools we discussed, actually annotate what protein, what genes, what are the genes in this certain genome, and then from those genes extract the potential fiber protein-like structure, whatever that is for that virus family, and use it to decide whether that particular virus isolate is going to infect human or not. And this will give us at least a way to cope with an emerging problem about an infection affecting the whole humankind in time, which is not a science fiction nowadays, because we have decent sequencing machines. Some of them are portable, that can probably sequence a virus genome, and then identifying it probably through the pipelines we have can be done. And if we can come up with good predictive models, this is not like a dream as one can imagine. So for this study, I just want to thank my students, Onurcan Karabulut. This was his part of his thesis, and Ahmet, Betül, and Erdem also contributed a lot. The funding for this project came from TÜBİTAK 2501 program project number. So I just want to take my funding agency and my students for this. We had roughly 4, so our publications relevant to this study is listed here. So if you're interested in the details or inspired from them, they are available through PubMed. My last few words, I tried to today provide an overview for virus in bioinformatics to the participants in the context, using the context of the vast virus world and my own research, my small piece in the community. And I'm hoping it was helpful and it was clear. Again, I'd like to thank to the organization for the invitation, giving me opportunity to speak to all of you, to all participants sparing their Friday evening just before the weekend. And again, my members of the SUSEK Lab for their support. With that, I can take some questions, and even if the time is not enough for questions, or if they don't come to your mind right now, you can email me. These are my emails. So offline or online, you can contact me. Right, thank you.
Of course, Isaac. Thank you sincerely for this enlightening workshop training. So if you wish, we can now transition to the question and answer session, which will consist of questions formulated by our team and those submitted by our participants.
So should I stop sharing the slides or continue sharing the slides? How do you work? I can keep them.
You can stop sharing. We're going to ask some questions now.
All right.
I'm starting with the first question.
Okay.
Should different feature sets be used for different virus species in virus host PPI prediction? Are virus species models more effective than a generalized model?
Well, certainly in the context of if you are doing protein-protein interaction, coming up with different feature sets will boost the performance. So majority of the researchers right now are working on, rather than algorithms, the encodings. They can come up with the features, they can come up with the enhanced performances. So your short answer to your first question is yes, certainly feature sets may improve the performance. The second question disappeared before I read it, so can you bring it up again? All right, so are virus-specific models more effective than a generalized model? So if you are asking this in the context of my own research, the parameter that would change if we bring another virus family to the picture, let's say if this was in influenza family, the only thing that will change will be the receptors on the host site and the mechanism of action, or the virus protein contributing to the entrance to the cell. So once those parameters change, the computation of individual PPI algorithms, the taxon information for the host will all remain same. So the beginning of the pipeline that will create the actual feature vector that will eventually be used to do the prediction for host source identification will remain the same. So this is not necessarily generalized model as in the sense that it's not that it's a generalized model, but there is a parameterization that involves to make it more virus specific. Now, when it comes to PPI prediction, protein-protein prediction, it's hard to tell because we cannot test it. Well, we don't have a lot of data to make sure that holds or not. But in literature, there are tools that separates the virus families, create training sets for individual virus families, and assess their performance for individual families. And it turns out for some families, they perform better than others. So if you look at that literature, the second probably has a basis for it. Maybe actually there are patterns in certain viruses that, whether through the feature extraction or encoding, that presents itself to the algorithm, and model is generated specific to that virus and it performs better. So although it became longer than the previous one, but again, short answer to your question is probably yes.
Thank you for your answer. The next question is from one of our YouTube participants. While the increase in virus species increased at a certain level between 1971 and 2014, why did it reach a strikingly high number of species after 2014?
It was strikingly higher. I mean, it caught my attention as well, but I didn't put effort to investigate it. But I can speculate on it, actually. There are a couple of dynamics that may be contributing to that. One could be the sequencing technologies may become more accessible around those times. So back in the day when I started bioinformatics, there were sequencers that were occupying buildings of buildings. So there are many buildings of sequencers. But now there are handheld sequencers. There may be a contributing factor on the technology's emergence, and not only emergence, but accessibility of the technologies, sequencing technologies. That might be one contributing factor. The other contributing factor could be there may be a rather bad outbreak around that time that may, again, just like we see in the last 5 years, all of a sudden increase the attention around viruses. So a lot of people started to work on viruses that emerged. It presented itself as a lot of new species. So that's my speculations here. But I mean, feel free to investigate. Actually, ICTV site has those numbers, and it's just on my slides. I have a timeline that tells when were the. Hold on. If you give me a couple of seconds, let me see what happened in terms of host shift in 2000. Maybe there was a unexpected thing in 2005 or 2014. All right, so in 2014 there was Ebola, avian influenza, and then loss of fever, and then another virus. But right before that there was MERS. So MERS is a pretty bad outbreak. So there is a accumulation of a lot of outbreaks, and Zika is around that time as well. And they experimented new sequencing technologies in Zika. I recall that I think they used this small sequencing machine, Zika. Probably outbreaks may be a contributing factor to it, along with technologies. Hope that answers. But it's worthwhile to investigate. And nice catching it. Thank you.
Thank you for your answer. The next question, again, one of our YouTube participants. What types of analysis do computational tools for virus taxonomy typically employ?
Well, so at species level, the reference genomes, my understanding, and I'm not a phylogeneticist, by the way. So at the species level, ICTV already identifies how the species look like. So at that level, the variance is higher anyways between species. So once you identify the species, though, once you go lower, what you only have in your hand is identification of the variation pool. So are there recombinations? Are they point mutations? And they, I don't know if it's the right term, but they have marker genes when they are calling the genotypes for certain species, certain viruses. So for instance, I'm just making up, L4 gene is going to be used for identifying serotypes for this organism is decided, virus decided. And then everybody looks at what kind of variations are happening in that segment of the genome, and then the calls of the clade or serotype or subtypes are based on that marker segment of the genome. So species is easy. Going down actually is all about the variations it contains. And in terms of computational tools, they are alignment tools. So we are talking about multiple sequence aligners, MSAs, or a variation of them, or pairwise aligners, if you have a reference to compare against. Alignment is the short answer.
Thank you for your answer again. The next question is from YouTube. The mimicry of host protein structures by viruses enables the the use of structural similarities in PPI prediction. Could this be a strategy involved by viruses to invade the host immune system?
The mimicry of host protein structures. I mean, I guess you have the answer in your question. Now, if the first part holds, I'm not an expert on this one, but I cannot answer correctly. But if the first part holds, obviously, right? So it's mimicking a host protein structure, so it can evade the immune system. No, if you are referring to what I said in terms of using virus proteins as a treatment protocol, if my reading is correct, what they are saying actually is it's so distantly homolog that even human immune system cannot recognize it, but it's functional. So it can't recognize it in a way that it will like attack it and use as a external entity. It just cannot recognize it. So it may be mimicry, but from what I read, is because of the distance in the homology is contributing to not attacking that certain proteins ortholog that comes from virus. If that's the case you are referring to.
Thank you for your response. The next question, again from YouTube. Are the currently proposed filtering methods for reducing false positive motive-based predictions sufficiently effective, or is a more advanced strategy required?
So the currently proposed filtering methods, so I'm not sure what Simge is referring to. Maybe we can have a follow-up explanation.
Yeah, if you want, we can ask the person who asked, then we will come back to the question.
All right. Yeah, I need a clarification of who proposes it and what are the filtering methods referred to.
All right. So moving on to the next question. It's again from the YouTube. Documenting all virus mammal interaction is impractical. So how can we predict host virus networks to map undiscovered viral interactions?
Well, that was our hope in our own research phase. So at least narrowing down the pool to potential interactions. If you are referring to infection in the context of interaction, but if you are referring to protein-protein resolution, that's even more impossible because we cannot test all mammalian kingdoms, all potential protein-protein pairs and virus pairs, and test them in a wet lab environment. All these tools, protein-protein interaction tools, or in my case, virus-host interaction or infection prediction or tools, are trying to extrapolate what we already know in a limited fashion to the remaining universe. And that's how we are probably going to cover undiscovered viral interactions unless there is a new biotechnological breakthrough coming that will be able to mimic or model these interactions in a lab environment at the single cell level or something like that. I don't know whether it's not my field, but if somebody comes up with a mechanism to do that, then it becomes feasible. But with the current technology, what we have is we have a limited base. We use it. We create models like machine learning models. We predict the rest. Hope the predictions are correct. But because it predicts the known well, probably it's going to do well with the remaining universe. Is where we are. And in many things where we are actually is this actually, because we cannot again test all the possibilities in lab.
Thank you for your response. The other question is again one for you, the participants. Most machine learning methods are designed for interspecies PPIs. What are the major methodological challenges faced by approaches specifically developed for various host PPI prediction?
Right. Ethem, now I didn't develop a PPI from scratch. So, but the starting point, now for one of my graduate students, we did a co-evolution based assessment and see if co-evolution can help in establishing a basis for PPI interaction. And it turns out co-evolution, meaning two interacting protein within a species evolving together, can be tested. And then the trees of evolution, through comparison of trees, we can predict with some accuracy whether they interact or not. So in that sense, when it's interspecies, there is an advantage of using co-evolution. When it is interspecies, and especially in viruses where even within the same host there may be many types of the same virus through mutations, even different cells may be resulting in different versions of the virus. It's hard to discuss a co-evolution. So rather than a opportunistic, random set of events contributing to interactions. So in that sense, in the absence of evolutionary piece, we only need to use the sequences themselves or structures or the effect of mutations on the structure to be able to decide whether that interaction still holds or not. And it's a big loss actually, because the phylogenetic trees gives a advantage in terms of pattern when identifying intra-species PPIs, but in the absence of it, it's a little bit a more challenging problem. So that's why probably none of the tools we have seen so far are consistently giving the similar results, but rather the results are all over the place. And on the top of that, actually, none of them are perfect. So it's a more challenging PPI prediction.
Thank you for your answer. This was our last question. So,
thank you.
Thank you very much for taking the time to share your knowledge and for your informative training on behalf of myself, our team, and all the participants.
Thank you for the opportunity. Have a wonderful weekend.